Good morning, everyone. My name is Alejandro. I'm a master's student at McGill University, and today I'm going to present my master's project. Combining gene genealogies and pedigrees to inform genetic screening programs, and my supervisor is the Dr. Simon Grabel. So there are around 7,000 different rare diseases, and 70 to 80% of them are believed to have a genomic origin. These subsets are usually caused by rare variants, and they're usually detected by sequencing, and imputation is challenging because most of them are not present on the reference panel that are used for imputation. However, there's a subset of them that has a high frequency in Quebec, and that's due to a founder effect. So let's talk about Quebec and the founder effect. So in the 16 and 1700s, around 8,500 individuals migrated from France to Quebec, as you can see here in one. And after that, they expanded through the Quebec territory, which causes successive founder events. So I'm going to talk about one of the most well documented ones, as you can see here in the plot. So they arrived in Quebec City, and then they migrated to Charlevoix, and then to Saguenay-Lac-Saint-Jean. And this is one of the most well documented, but it's not the only one. There are other ones. And the result of these successive founder effects is an uneven distribution of allele frequencies across the Quebec territory, and that causes an increased regional frequency of certain disease-causing mutations. And it is really important to identify the regions where the frequency of this disease-causing mutation is higher to inform the genetic screening programs. How do we know so much about these founder events? Is because we have genealogical records available through the Balzac project, which was it's a huge genealogy for French Canadians in Quebec, which has around 6 million individuals, and that was reconstructed from marriage and death certificates by the Catholic Church. So how can we identify these regions with a higher frequency? So I have an example here with this rare disease, which is chronic atrial and intestinal dysrhythmia. So a PhD student in Simon's lab with clinicians, they identify 11 cases that all share the same mutation in the SGO1 gene. And they perform an haplotype analysis of these 11 French Canadian cases, and they realize that they all share a long haplotype of 2.9 megabases. This mutation has been found in Europe. So they compared the haplotype from the French Canadians with the Swedish case, and they realized that this Swedish case share a small portion of this big haplotype. So they assumed that this mutation was introduced into the Quebec population uh, once, so a single introduction, and they developed a new algorithm called EastGen to from a set of probands in a genealogy to find a common ancestor, which is the person that introduced the mutation, and then with that founder, you can perform allele dropping simulations to estimate the carrier frequency. So they estimated the carrier frequency in different regions of Quebec, and as you can see here in this map, this variant has a higher frequency in Charlevoix and Saguenay, which is concordant with the founder event that happened in that region, and the frequency could be as high as 1 over 155 individuals. So this is great, but the problem is that it takes a lot of time to do this for every mutation. So how can we improve this process with the data that we have available right now? So Quebec has its own biobank, which is the Cartagena cohort, which has 43,000 individuals. 30,000 of them have genotype array data available, and a subset of 2,000 individuals have whole genome sequencing data available. Moreover, we have access to the Balzac genealogy, which has 6.5 million individuals from the founders from the 1600s to the present day individuals, and 8,000 of them are from Cartagena. So with this, we can link genetic and genealogical data. And also we have the ancestral recombination graph with recent improvements by Palomar Lab in Oxford that they developed ArcNeedle, which allows the use of genotype data to reconstruct these mathematical objects. So I'm going to focus a little bit more on the ARC because it's important for this project. So if you want to study the transmission history of a mutation, you can use a genealogy. So here we can see an example with 3 different regions in the same chromosome, and you can see how they transmit generation by generation. And the recombination is going to change the transmission patterns a little bit. However, for biobank scale data, we often don't have genealogies. So what you can do is that you can take the genetic data you have, for example, genotype array data, and you can try to reconstruct these gene genealogies using the haplotype sharing information that you have. So here we have the same three regions, and we have three highly correlated trees. The only difference between them is that if there's a recombination, that's going to break the haplotype and it's going to change the nodes a little bit. But the important message here is that this means that we can barely have one tree per mutation, and we can use that tree to infer a lot of information. So the goal of this project was to develop a new method using the R that can quickly assess if a mutation was introduced one time or multiple times into the Quebec population, and also use the tree to perform genotype imputation of rare variants to increase the sample size. So for this, we use the genotype array individuals from Cartagena, and we use R-needle to reconstruct the arc. And we use the whole genome sequencing individuals to detect rare variants. And here I'm going to focus on 9 non-founder mutations in the Saguenay-Lac-Saint-Jean region, because we want to validate our method with known founder mutations. So we're going to use the ARC and the carrier status to perform genotype imputation, and also to assess if the mutation was introduced once or not. And then we're going to use ISGen and the Balzac genealogy to estimate the carrier frequency at a really fine scale. So here I'm going to explain a little bit how the imputation and the timing works using the ARC. So let's say that you have two individuals that have a rare mutation that you identify by whole genome sequencing. We also have two individuals that are negative. They don't have the mutation, but we have a few individuals in the tree that they have genotype array data, but they were not sequenced. So we don't know if they're carriers or not. So what we're going to do is that we're going to take these two carriers, and we're going to find a common ancestor in the local tree for that mutation, which in case is this one. So
So
we have a time T1, which is the mutation age that we're going to use. And we're going to say that all the individuals below that common ancestor are going to be carriers because they share a long haplotype. So here you can see that that individual in the middle now is going to assign as 1, which means that it's a carrier. But there's going to be some individuals that are going to be between carriers and non-carriers. And for those one, we're just going to assign a posterior probability that is going to be higher if it's really close to the common ancestor. So in this case, it was 0.7. This imputation is really similar to the one proposed in the R-needle paper. So now I'm going to show you the results. We analyzed 9 mutations that causes rare diseases in Quebec. So here we have mucolipidosis, Zellweger syndrome, tyrosinemia type 1, among other ones. And we computed a correlation between the nomad frequency for non-Finnish European and the mutation age that I explained earlier. And what we can see here is that there's a correlation between these two. So variants that have a really low frequency in nomad for non-Finnish European are consistent with a lower mutation age, and therefore a single introduction into the Quebec population. And then we estimated the carrier frequency using 3 different methods. So we have the whole genome sequencing data, we have the ARC imputation, and we have ISGen, which is a really fine scale and uses the genealogy. And we compare that against the literature. So what we can see here is that, for example, for the FAH variant that causes tyrosinemia type 1, there's a good agreement between all the metrics. So either if you use only the whole genome sequencing data, or you impute with the ARC, or you use ISGen, there's a really good agreement between these carrier frequencies. And in some cases, like this variant that causes late syndrome French Canadian type, you see that the whole genome sequencing estimate is different from the rest. That means that sometimes when you have a low number of carriers in your whole genome sequencing cohort, you don't have enough power to actually estimate the carrier frequency, and that's why you should use imputation. And we perform a collaboration with the National Institute of Excellence in Health and Social Services of Quebec, where we analyze variants that cause Zellweger syndrome. And we analyze one variant which is located in the PEX6 gene. As you can see here in this map, there's a higher frequency in Saguenay-Lac-Saint-Jean, Charlevoix, and Côte-Nord. So this institute released a report, and they used this as one line of evidence, and they recommended adding this variant to the carrier screening testing for these 3 regions. And finally, I want to talk about the application of the ARC to Colombian population. So we have this case report, which it's a patient that has Eurofacial syndrome that was discovered by the Dr. Ochoa in Medellin, Colombia. So we wanted to perform genetic diagnosis. So we performed whole exome sequencing and we analyzed the FASTQ files. We did the standard bioinformatic analysis. And then we used the BCF files to perform variant prioritization. And we found that this patient was homozygous for a variant in the HPS E2 gene, which has already been reported before in patients from Colombia with Eurofacial syndrome. There was a haplotype analysis suggesting that this might be a founder mutation. So we decided to, and we published this year, so you can go and look it up. So we decided to use the ancestral recombination graph to confirm if this was a founder mutation or not. So we took the BCF file from our case and we merged it with the BCF files from the Colombians living in Medellin from the 1,000 genomes population. We performed genotype imputation using the TopMed imputation server. So we ended up with around 500,000 variants. And then we reconstructed the ancestral recombination graph using RNeedle, and we analyzed the haplotype and we estimated the mutation age of the patients. And what we saw is that the two haplotypes carried by this patient, they share a 7.2 to 7.4 megabases haplotype, and the estimated mutation age was from 260 to 360 years ago, which is consistent with the colonization of the European individuals. So we wanted to know the ancestral origin of this haplotype. So we performed local ancestry analysis using RF mix and using Yorubas as African reference, Native Americans from the HGDP, and also Europeans for this analysis. And we actually found that this mutation has an European, this haplotype has an European origin, as you can see here in this plot for the chromosome 10. Here is the location of the gene, and is contained in an European haplotype for both copies. So as conclusions, the ARC captures the transmission history of rare variants, and we can combine the ARC with genealogical data to get a really accurate carrier frequency. Also, the ARC is useful even in admixed populations where founder effects are important, like Colombia. You can analyze mutations in admixed populations. And as limitations, we're using EastGen for the carrier frequency estimation. That's quite slow, takes 4 days per mutation. And for some of these analyses, you will need genealogical records. Finally, I want to thank Simon Gravel's lab, our collaborators that help with the validation and the data for this project. Also for the Ochoa syndrome, I want to mention Joanna and Wildeman. And I also want to thank Mytax and the University of Antioquia. And if you're interested in reading the how we can use the R for admixed populations with the case reports, we have a QR code here for our paper. Thank you.